everyone to the first CACNA1A Family and Scientific Conference. My name is Lisa Manaster, and I am the president and a co-founder of the CACNA1A Foundation. This is incredible. Words really can't express how special today is. 1 year ago, a family foundation for CACNA1A related diseases did not exist. And here we are in a virtual room with over 200 registered attendees. Well, they're not actually on right now, but they're registered. From the United States, Canada, Mexico, the UK, Australia, New Zealand, Austria, Denmark, Germany, Greece, Italy, Turkey, I'm going to keep going, Georgia, Poland, Russia, Hong Kong, Japan, Malaysia, and Singapore, all ready to engage with the experts in our disease. I am in awe of what we have accomplished, and I want to thank you all for being a part of this journey. We could not have done it without you, the patients, parents, and caregivers, family members, friends, scientists and researchers, the clinicians who see our patients daily, and industry members. Like all the parents here today would say, I have an amazing, hardworking daughter, but she struggles with epilepsy, ataxia, nystagmus, migraines, daily headaches and dizzy spells, intellectual disability, and a host of other issues. We had no idea why. It was not until 3 years ago, when she was 20 years old, that we finally received a diagnosis of her CAC-NA1A variant. Parenting a child with special needs can be isolating, lonely, stressful. There are so many adjectives I could use, as many of you know, not all of them negative, because I also feel extremely blessed to be her mom, and I'm so, so proud of her. But when I turned to her doctors for advice and they couldn't help me answer my questions, that was really hard. But now we have an organization just for us. I want you to know that the CACNA1A Foundation is working hard to build an engaged, patient-led research network. Your being here today sends a message to the world that we need to find treatments and a cure for those suffering from CACNA1A related diseases. We thank you for taking the time this weekend to show your support for our loved ones. We are so grateful. Before I continue, I want to take a moment to acknowledge our conference committee, Sunitha Malapati, Amy Jung, and Helena Riley for their help in planning this weekend's events. We'd also like to shout out to Jeff Lord from My City Med for all of the behind the scenes work on the conference platform. We had a hardworking team, and hopefully it all goes off without a hitch. Next, I'd like to thank our sponsors who generously supported this event to make it possible. Prostate Centers, Potomac Urology, Greenwich Biosciences, Ovid Therapeutics, PRC Saltillo, and Napa Neurological and Physical Habilitation Centers. Most importantly, I must thank our board of directors and scientific advisory board members who are the driving force behind the scenes, giving so much of their time, their hearts and souls to ensure our success. Allison Buckner, Carolyn Anderson, and Sunitha Malapati, who you'll all meet over the next 2 days, and doctors Henry Colcraft, Christopher Gomez, Joanna Jen, Jeff Nobles, Suki Ko, Elsa Rosignol, Michael Wangler, and our newest board member, Charlie Cohn. I also want to acknowledge and thank Dr. Wendy Chung, who has developed and spearheaded our natural history study, which, as you'll hear shortly, is one of the most critical elements for finding a cure. Finally, I'd like to thank Shanna Talbert for her hard work, which has been invaluable to our success. So this is on our website, but I'd like to take a moment to read you the vision and the mission of the CACNA1A Foundation, as it is the driving force behind everything we are doing. Our vision, of course, is a world free of the debilitating effects of CACNA1A related disorders. And our mission, how we hope to get there, is by finding specific treatment options and a cure for CACNA1A patients by building a collaborative network of patients, families, clinicians, and scientists that will all work together to raise awareness and accelerate the understanding, diagnosis, and treatment of CACNA1A related diseases. Collaboration. We believe that's the key to our success. There is incredible research going on all over the world on our gene, but it tends to be siloed within specific labs. In order to improve diagnosis and prognosis, the CACNA1A Foundation hopes to facilitate collaboration and the sharing of knowledge among researchers. But I'm probably getting ahead of myself. First, I'd like to touch on some of our accomplishments this year. This timeline illustrates some of our key milestones. We received our nonprofit approval last May, launched the website almost 1 year ago on August 6th of 2020, and by the end of the year had raised just shy of $200,000 for research. We launched a natural history study in January of this year, and in March, funded 3 $50,000 research grants. The recipients of those funds will be speaking tomorrow about their projects. I do hope you'll be able to tune in. We hosted our first awareness day, I should say the first CACNA1A Awareness Day in March, hosted a 5K that raised an incredible $70,000 for research in June. And of course, here we are today at this conference. Next week, we will be announcing our biobanking program and will continue to fund cutting-edge translational research. It's been a busy and productive year. I have to say, we couldn't have accomplished so much without the help of so many partners. These organizations all share in the ultimate goal of identifying, treating, and curing rare diseases. We arrived on the scene, they embraced us, and have mentored us on this journey. We are so grateful for their collaboration, guidance, and support. But there is still so much to do. First and foremost, this year, we plan to lay the groundwork so our disease is clinical trial ready. This entails having robust natural history study data, a library of biospecimens and cell lines, and understanding and identifying endpoints and biomarkers for our disease. We also plan to announce a partnership with RareX for the CACNA1A data collection system, begin to develop a comprehensive database that includes functional characterizations of variants to understand how they act on the channel. So are they a gain of function or a loss of function? Even of mixed function, we're discovering that too, which will inform science and advance treatments. We plan to continue to increase awareness of CACNA1A by attending conferences. We actually attended the American Epilepsy Society this year, and we have a few more lined up for the coming year. And finally, we need to engage stakeholders and develop relationships with biopharma. As an organization, we are striving to be at the forefront of all things CACNA1A. But really, the only way we can succeed is if we have an engaged patient population. We are stronger together, and we need you to participate in these initiatives. So if you have not already, please join us. We send a monthly newsletter and post on social media. Our blog is a source of timely information. We say it again and again, but please fill out our natural history study and our soon to be launched data collection system, which you'll hear more about in the near future. Send us your blood samples. It's going to be really easy to do, we promise. We are excited about the opportunities ahead and are optimistic about our children's futures. We hope you are too. So at the bottom of the slide is my email address. Please feel free to reach out to me if you have any questions or want to help in any way. But for now, sit back, enjoy listening to our impressive lineup of speakers. My hope is that you take away from this conference a few things. You learn more about our disease and what needs to be done to find a cure, and that you leave this conference feeling empowered, that you can truly help make a difference, because you can. Okay, so next, I'd like to introduce Dr. Tracy Dixon-Salazar, our keynote speaker. We are honored and grateful that she has agreed to speak today. Dr. Dixon-Salazar is a neuroscientist, geneticist, patient advocate, and the executive director of the LGS Foundation, a nonprofit organization dedicated to providing information about Lennox-Gastaut syndrome, a rare and severe form of childhood onset epilepsy. Many of our CACNA1A children have a diagnosis of LGS, so you may be familiar with this rare epilepsy. As executive director, Dr. Dixon-Salazar executes the foundation's strategic vision that will one day lead to prevention of and cures for LGS. Her desire to get her PhD was inspired by her daughter, who developed uncontrolled epilepsy at the age of 2, which then evolved into Lennox-Gastaut syndrome by the age of 5. After 16 years of watching daily unrelenting seizures in her daughter, she uncovered the genetic driver of her illness and identified a novel precision therapy that improved her life. She has more than 20 years of nonprofit leadership and advocacy experience. She believes in the importance of targeting both the causes and symptoms of disease and is working to reshape the future of medicine so others in rare disease may go farther, faster, which is great news for us. Welcome, Tracy. So I'm going to stop sharing.
Thank you for that lovely introduction. Yeah, I really never know what to say. I never know if I'm wearing my mom hat or my scientist hat or my advocate hat. So thank you guys for having me. I'll start sharing in just a second. I just want to say that this talk is for really for the families out there. I was asked by Lisa to come here and and help to emphasize how important patients and families are in the medical journey. And I remember when I got started in this journey a long, long time ago, I was like, well, what could I possibly know? What could I possibly bring to the table? And now, fast forward, we're on our 26th year of our journey with LGS and rare disease. And patients should be involved in every aspect of research and care. And I hope that the story that I'm going to tell you today, the journey that my family has been on, will help you to see how far we've come and also know how far we need to go and know how it's going to take each and every one of us screaming at the top of our lungs to change a healthcare system that doesn't want to change. So, I'm excited to be here, and this is really for the families, for all of my doctor colleagues and friends on this call. I hope you hear this message with the love and the compassion and the desire for change that I want to see. And I know I'm preaching to the choir. All of you here are the physicians and researchers who already get that, but there are so many out there that really don't value the patient voice. So I'm going to talk about, I love this concept of further, faster. I think if you get involved in research and what you've accomplished so far, Lisa, has just been nothing short of amazing. You and your team of dedicated parents and families and scientists and clinicians. So congratulations. But we will go further faster if patients are part of the process. And I'm going to talk a little bit. I am the mom who got a PhD to understand my kid's disease. And hopefully at the end, I'll answer whether or not you actually need to get your PhD in order to understand your loved one's disease. So I think many of you know about LGS, but let me just show you, if you were to ask Dr. Tracy or Scientist Tracy what LGS is, you'd probably see a slide that looks something like this. LGS, in order to get the diagnosis, you need to have multiple seizure types. Tonic seizures are usually present, and then a number of others. Age of onset of seizures is usually under the age of 7, with diagnosis of LGS being most common between the ages of 3 and 5 years old. A very specific EEG pattern, very specific biomarkers are associated with LGS, and you can see two of those over here. And then cognitive impairment is not required for the diagnosis of LGS, but it is present in nearly everybody within 5 years of seizure onset. And that's what you would see. And a lot of times a doctor or a scientist will open a textbook and they'll see this and they'll say, oh, this is a pediatric onset disease. I'm an adult neurologist. I don't have to think about this. But these kids do grow up to become adults. And I've had that conversation many a time, that it is not just a pediatric disease. It's pediatric onset disease. But none of this definition takes into account the journey that we have been on as families. And I want to tell you a little bit about our journey. So this next slide contains seizures of my daughter having seizures. And so if you want to look away, if you're sick of seeing seizures, please do. This is what our introduction was to the world of LGS. We were awoken in the middle of the night to our daughter shaking and gagging. We had no idea this was a seizure. We thought she was choking. She was blue in the face, turning white, not getting oxygen. Her lips were white, and she was stiff as a board. And twitching. She had peed herself. And I remember that we felt completely helpless. We didn't know what to do. We called 911 and they came, and by the time they got there, this was over. And this was not the first seizure. This just is one of the ones that came later that looked like the first seizure. And he said, I'll never forget what he said. He said, you know, her airway is clear, but what you just described sounds a lot like a seizure. And I remember our response was, what's a seizure? And this was just the beginning of what would be thousands and thousands of seizures over the next 16 years. We felt so helpless. And really, if you don't have a loved one with epilepsy, all you have to do is imagine for 1 second what your life would be like if you or someone you love could drop at any moment and have a seizure. So myoclonic seizure. Savannah very quickly began having multiple seizure types. This would be atypical absence, myoclonic astatic. I don't even know at this point, and I've been doing this for a long time. But her journey really began with this harsh introduction, and then there was this evolution that nobody ever talked about. So here she was, born in 1993, and she was perfectly typical, developed beautifully for the first couple of years. She was not delayed, she was not precocious, and then there was that first seizure. And doctors didn't call them seizures at the beginning. They called them spells and episodes. And I later learned it was because they didn't want to label her with this awful word called epilepsy. So our introduction to the stigma of epilepsy was actually brought to us by the physician who didn't want to label her, which is really interesting. To no fault of the physician at all. I mean, he was just doing the best he could, but it's looking back, you know, hindsight is 20/20, right? And it's like, oh, interesting. At age 3, she was diagnosed with intractable epilepsy. And back then, and thank God we don't call them these things anymore. And if you're still calling them these things, please stop it. We didn't know if she had idiopathic or cryptogenic epilepsy. And so later on, you know, maybe 10 or so years into our journey, we learned that idiopathic meant presumed genetic and symptomatic meant structural. So you saw something on the MRI, and then cryptogenic meant unknown, intractable epilepsy. And we actually learned about this by reading her medical record, not that we were ever told this. We learned it by reading her medical record. Again, to no fault of the physicians that treated her, did a great job, but often, and I've learned this over my many years in the space, is that we learn something at the scientific level and at the clinical level, and it never makes its way down to the patient or to the patient community. And that's just one reason why it's so important that we're involved, so we can be bringing that information rapidly to our families. She started having more than one seizure type at the age of 3. At one point, she was having 6 different seizure types. We didn't know if they were grand mal or tonic-clonic. At some point, you know, they changed the name of grand mal seizures to tonic-clonic. And again, that didn't trickle down to the patient community. So it really impacted our ability to understand what we were talking about, right? It was very confusing. And then it wasn't until the age of 5 that she actually got her diagnosis of LGS with this triad of symptoms, as they called it last year. Back then. And we now know that it's more than 3 symptoms, right? Because you need to have age of onset in childhood too. So it's a quadrad or whatever it would be. But I thought she was misdiagnosed up until that point. Oh, she's had LGS this whole time. But we didn't know it. Nobody had diagnosed her with it because we hadn't gotten a good EEG up until then. But that's really not what we believe today. And we found out that she had LGS because she went in for a brain surgery workup, which she wasn't a candidate for. Otherwise, we would have never known. Video EEG monitoring, I'm so happy that's standard of care now, but it was not back in the early 90s. And really what we knew about LGS was that it was this umbrella disorder that could evolve from a number of different things and a number of different causes. But these 2 pictures, this umbrella picture and this 3-dot picture, is really what we knew about LGS back then. Savannah went on to continue to have many, many seizures, and a paper was published a few years ago about the treatment loop in LGS. I think this paper is maybe probably 8 or 9 years old now, but we were in this treatment loop where you go from, oh, your surgical evaluation, can you get resective surgery? No. How about palliative surgery? How about a corpus callosotomy or a VNS? And if that doesn't work, you can go try a medicine. And there's really no shortage of medicines that we can try, but as is the case with the majority of our families, you try, and nothing stops the seizures. And then new treatments are coming out all the time, which thank God they are, because otherwise people would just be saying, gosh, I'm really sorry you're having seizures all the time and that your life is an utter and complete hell. But we're not going to make any new medicines. But I also hope that we're making medicines in a different way, you know, 5 to 10 years from now. So we continued on this way for 16 years, and Savannah had more than 40,000 seizures. She tried and failed more than 26 different therapies. And I, as a parent, continue to remain exceptionally confused about what LGS meant. And it turns out in my learnings through my PhD and my work with advocacy, that the definition of LGS has changed at least 6 times. There's actually a new proposed definition out right now. And so sometimes it requires this EEG pattern, sometimes it doesn't. Sometimes you have to have tonic seizure, sometimes you don't. And again, this is no fault of the medical system. This is how science works, right? You learn new things, you change names, but it's really confusing for the patients. And it also impacts my ability to get services for my daughter. Every time a name change is made up at the top. And I think, one example is atonic seizures versus drop seizures. It used to be atonic seizures meant, the body would go completely limp, right? So a loss of tone. But then drop seizures came on them, came in basically from what I can tell through industry and the FDA, who wanted to sort of group any seizure that made you fall. But we had previously called drop seizures, atonic seizures, drop seizures. And then now we've got the focal epilepsy versus the partial epilepsy distinguishing. And I still on a daily basis hear grand mal from the community members that we work with. So, these changes are really difficult. So it's so important for us to be gathering this healthcare knowledge, this literacy, and bringing it back to our families, because across my journey and probably across yours, it doesn't always make it down after the scientists and the clinicians have discussed it. So this has really been a difficult journey for us. We've had a different story with each new doctor. We were always thought of as a common disease, epilepsy, and not as a rare disease. Another way to snapshot Savannah's journey is like this, to say, okay, during this 16-year period from 1995 to 2011, she had more than 40,000 seizures. It took her 3 years to get the LGS diagnosis. It took her 7 doctors before we got it. 26 treatments failed, lots of hospitalizations and surgeries, and a very, very expensive medical odyssey. But again, that doesn't capture the journey. And to this day, I don't know that science is really capturing the journey. And I may be one of the few that's out there actually talking about LGS in this way, but here's Savannah's journey. So she had an etiology. All of us have a cause. Our loved ones who have seizures have a cause. We may not know it. We didn't know it for many, many years for Savannah. For 16 years, we didn't know it. But everybody has an etiology of some kind, and whether it's a brain injury or an early life, or it's genetic, or it's something else. And then that leads to the first seizure. And so for Savannah, her first seizure was at 2. And then she developed epilepsy, which essentially very general definition is 2 or more seizures. Often people will say 2 or more unprovoked seizures, but all seizures are provoked by a drop in seizure threshold. So I struggle a little bit with that term, but 2 or more seizures. So she had she's had her first seizure at 2, and then 4 seizures over the next 2 months. And then she went 6 months without having any seizures. So she had this quiet period. And then when she turned 3, the seizures came back with a vengeance. She started having multiple seizure types, hundreds and hundreds of seizures a day. A good day was 5 seizures. A bad day was like 500. I stopped counting. And the real kicker was that you never knew what you were going to get. Is this a 5 seizure day? Is this a 500 seizure day? Are we going to be resuscitating her? Are we going to be in the hospital? Is she going to live? It was an exhausting 16 years. At age 3 is when developmental delay also became apparent for Savannah. And then the LGS diagnosis at the age of 5. And let's just call it what it is. The abnormal EEG with LGS is the sign that something has gone horribly wrong with brain development. You have very, very abnormal brain waves. That is a sign of really horrible brain damage. And I think it's important to say that, because back in the day, we were told many, many times that seizures do not cause brain damage. And yes, lots and lots of seizures do cause brain damage. I want to point this out, and this is going to come up a little bit later in our journey story, is that across our LGS journey too, when she is of age of 5 and she had the surgery workup and wasn't a candidate for surgery, we put her on the ketogenic diet. And the dietitian said she needs to go on calcium supplements and vitamin supplements in general. And I said, okay. And she's like, and at some point in the journey, I'll have to go back and map this, but it became very important to put kids with epilepsy in general on calcium and vitamin D supplements, because the seizure meds are very hard on bones. And so we put her on for the first time with the ketogenic diet, this calcium supplement and vitamins. And we lost control with the diet. We had seizure control with the diet at that point for several months, and kept saying, do you think it's the vitamin? Do you think it's the vitamin? And the doctor was like, no, it can't be the vitamin. And we didn't have enough, thoughtfulness to think, oh, we should maybe try taking the vitamin away. And then over the next few years, we had 3 different trials of giving this kid calcium supplements. And every time she would end up in the hospital in status epilepticus. And it just never occurred to me or anybody that this was a sign of things to come. So this is going to come up again. But this was about the time that I went to college, when she was 5 years old, and got right around the time she got the LGS diagnosis. And I joke a little bit now about it, but I was such a young mother. I think I was 23 or 24 when I went back to college. And I joke, I was trying to read these papers. I'd go check out internet time at the library, and I try to read these scientific papers and these medical papers. And I'm like, I have to go to college because I don't understand English. I've never been to college. I didn't do that well in high school. And I need to understand English. And so I enrolled in the junior college and took a few English classes, and then realized that I needed to take some science classes, and then realized that those papers are not in English. Science is a different language, and medicine is a different language, and science and medicine are not the same language, right? And so we all have to learn this vocabulary so that we can communicate with each other. Doctors have to learn to speak researcher, researcher have to learn to speak doctor, everybody needs to learn to speak patient, patient needs to learn to speak everybody. And so that's another reason that I think it's really important, especially if we want to go further faster, is that we need to be communicating with each other and learning the languages. You know, I've never understood why they needed to call something symptomatic when they could call it, you know, structural abnormal MRI, right? And then we could all understand the language, but there are lots of medical terms that we need to understand. So I want to just fast forward a little bit. I'm going to go back to my journey in just a second, but I want to tell you what we know about LGS now. And in some ways, I feel like the LGS Foundation and patients and families are driving this conversation, but there is data in the literature that's still somewhat considered fringe. It's starting to get more and more prevalent. And I can remember many times talking about this evolution across my many years with researchers, and they're like, yeah, but most people's LGS or epilepsy, when they start having epilepsy, doesn't evolve. And I'm like, but some do, right? But anyway, so here's what we know now. And there's actually some evidence in the literature, some really nice research so for the etiologies of LGS, there's actually some preliminary small studies, natural history studies that say structural for about half of all LGS cases. So with birth injury, lack of oxygen at birth, hypoxic ischemia being really prevalent cause of seizures that evolved to LGS. And you also have genetic, about 15% of cases, and this is, as far as I can tell, more than 150 genes. We've got CACNA1A kids that evolved to LGS, SCN2A, SCN1A, Duke15q, trisomy 21. I mean, the list is very, very long. And then these other causes, metabolics, infectious, autoimmune. They develop their seizures mostly before the age of 8 years old. And then treatment-resistant epilepsy can evolve from other syndromes. And then now we have this new criteria from the International League Against Epilepsy that in order to have the diagnosis of LGS, tonic seizures associated with sleep, slow spike and wave on the EEG, paroxysmal fast activity on the EEG, an abnormal EEG background, and one other seizure type. So another definition. It really doesn't matter where you came from though. Once you have LGS, your prognosis is pretty awful. More than 85% will continue to have seizures into adulthood. More than 99% will have tonic seizures in sleep or associated with sleep stages. More than 90% will have significant cognitive impairment, increased risk of death. Most kids are diagnosed between the ages of 3 and 5 with LGS, but they do grow up. My daughter is 28 now, and we have people in our LGS community that are in their 40s and 50s and 60s. But over this evolution, you go from a normal brain network. You know, as my training and PhD as a neuroscientist, we learned that cells that fire together, wire together. And during this time in brain development in these really young children, the brain is developing and wiring. And then you have all of these abnormal electrical bursts of activity, this abnormal firing, and that changes the wiring. So by the time you have LGS, you have a very abnormal wiring pattern, and emerging evidence suggests that is what is responsible for the same EEG. No matter what your etiology is, you evolve to the same biomarker, and we need to understand the science of that. So I'm going to show you just one piece of data. If you get a chance to read this paper, this is a fabulous paper. This was a 10-year follow-up study where they followed a bunch of children who came into the tertiary medical center. It was done by Ann Berg and her colleagues. And they followed them for 10 years. So when they came in under their initial diagnosis, there were 4 patients that had another epilepsy diagnosis, 6 that were undiagnosed, 4 that had LGS at the time that they first came in, 23 that had West syndrome, 10 that had generalized epilepsy, and 11 that had focal epilepsy. 2 years later, you can see how many of these red lines went from where they were to becoming LGS. We went from 4 who had LGS to 22. So, and this usually happens within over a 2-year period, this evolution in really young children. So LGS is something that happens while we are sitting there watching our kids have seizure after seizure after seizure. And I am trying to argue that it is crucial for us to address the EEG features of LGS and start to think about pre-LGS and preventing the evolution to this horrible, horrible, rare epilepsy syndrome. So I'm going to go back to my journey now, and I know I'm jumping all over the place. I'm still trying to figure out how the best way is to tell this story, but I hope you guys are following along. But many of you guys have seen this, and I don't think the CACNA1A gene is on here. I'll need to put it on here, but this is the gene discovery and epilepsy timeline. So there were a few genes that were known. So this is when I started school, 1997. This is my associate's degree, and you can see here, here's my bachelor's, my PhD, and then my postdoc. So I started my postdoc right at the genomic revolution, right? So when I was getting my associates, I worked in a genetic lab, and we were sequencing one gene at a time, right? A couple of exons at a time. By the time I came back to my postdoc in genetics, I did my other training in neuroscience. By the time I came back, we were sequencing whole exomes. So approximately all 20,000 genes, or most of them. The world was just exploding in all of this. And it was an exciting time to be in the lab. I feel like, I mean, when I started going to college and really decided to stay in, I felt like I wanted to find the causes of epilepsy. Because when Savannah was first evolving and going along her journey, doctors would say to us, well, thankfully, we don't need to know what causes epilepsy in order to treat it. And yet she would fail treatment after treatment after treatment. And so when I really dedicated my career to becoming a researcher and wanting to just be a part of the community of people that were helping the next generation of Savannahs out there, I wanted to find causes. And so this was I had one teeny tiny piece of this. I published a number of papers on exome sequencing and finding causes of epilepsy in children. We had a number of CACNA1A families and other genes that really mean a lot to me that were later published to be actual families with genes, and we had sequenced them. So it's really exciting to me to think about that piece of the journey. But during this whole journey of all of my academic career, really, Savannah was not doing well. She was, by the time she was 18, she was the mental age of about a 2 or 3 year old because of all the seizures. And I had already said she tried and failed 26 different things, epilepsy drugs, diets, neurostimulators, devices, alternative therapies. She was having on average about 300 seizures a month. She was on 7 anti-seizure medications, plus a vagal nerve stimulator, plus a special diet. And she would go into cluster seizures and non-convulsive status every week. So she was getting rectal diazepine, benzodiazepines 2 to 4 times a week. And when that didn't work, we would go to the emergency room and get intravenous Ativan. So we get more. And when that didn't work, she would be put into a medically induced coma. And she spent most of her time asleep and unconscious, and it was really impossible to learn. And I remember talking to my postdoc advisor, Joe Gleason, one day, and he asked me how Savannah was doing, and I burst into tears, which is probably the worst thing you can do in front of your postdoc advisor or any of your mentors in the science community. But it led him and I to have a discussion about sequencing Savannah's exome. And this was really unusual back then because we were not sequencing sporadics. We did not know about de novo mutations. We still believed that genetic meant inherited. Genetic does not necessarily always mean inherited anymore. You can have de novo mutations, and it's a complex relationship that I won't get into, but it was really unusual to sequence somebody who had no family history of epilepsy. We had been sequencing families that had a significant history prior to that. So I'll jump to the the conclusion after many years of work. And so we identified a number of high impact variants in calcium channel subunits. And I want to remember back to that. Every time we put her on calcium supplementation, she would seize more. So we begin to look into these variants. We begin to think about that history that she had. We begin to dig into what these variants did. And here's the list of the variants. So you are not going to see CACNA1A on this list. She does not have mutations in that gene. And we're actually resequencing her right now and hopefully getting her whole genome back sometime this year to reaffirm this data, because this data is quite old. But she had a really high number of mutations in L-type calcium channel genes. And she has a huge de novo mutation load. And so I don't know if she was exposed to something in utero or what, but even we know that patients who have de novo mutations in the rare epilepsies and the severe epilepsies of childhood have a high mutation load anyway, but Savannah has a really high mutation load and in L-type channels. And we were desperate, okay? The kid was not doing well. So I started to dig in and do research on if there were any drugs that might target L-type calcium channel receptors. And a number of the variants suggested too much calcium was going into her cells, and the dietary supplement issue suggested too much calcium. So we ultimately found a drug, Varapamil, which is an L-type calcium channel blocker. And we're very lucky that it was too much calcium going into the cells and not the opposite, which would be too little, because there are not medicines necessarily, or there weren't back then. I don't think there are now, but there might be. That would increase calcium flow. So we were lucky that this drug existed. So we made a case. My postdoc advisor is also a pediatric neurologist, and two of us went together to talk to Savannah's treating neurologist and convinced him that Varapamil was something we should try. And within 2 weeks, we saw a huge reduction in seizures for her. And she ultimately stabled out at about a 95% reduction in seizures. And I just should mention that Varapamil is not FDA approved for seizures. It's used for arrhythmia and high blood pressure. But I think even more importantly on our quality of life and on Savannah's quality of life and on just her survival was that the rescue med use, the use of emergency rescue meds to stop her seizures just dropped. It just stopped within a couple of months. And I'm happy to report that we are now in year 11 of this really amazing journey of hers. And I just want to show you a short video clip of her.
It tickles. Tell you can. It tickles. This is Savannah. She doesn't spend her day seizing, unconscious. Bye-bye.
So Savannah's doing really, really well. And if you had told me back then that treatment number 27 was going to make the darndest bit of difference in our lives, I would have told you, no, she's got too much brain damage. The damage is done. Plus, none of the other 26 have worked. So why is 27 going to be the magic bullet? And it is not perfect. She is not seizure free, but this 99% reduction in seizures and this 100% reduction in status epilepticus and clusters has changed her life. She's come off 3 of her anti-seizure meds and her VNS. She's talking, she's awake. And I feel like I got to know her again for the first time at the age of 18. She's funny. And she's sassy and she's a smart aleck and she loves slapstick humor and, and she knows what she wants in life and is very, just outspoken. And I just felt like I got to know her for the first time. And so all of this really led me to become a big proponent of this genomic and precision medicine concept that is just prevalent today. And you guys are enacting this. The CACNA1A Foundation is doing this. You guys are going to move forward and try to find treatments that target the specific epilepsy and the specific issues and the headaches and all of those issues that are related to the cause. And that is huge because just 26 years ago, we didn't need to know the cause in order to treat the epilepsy was the mantra. You know, 26 years ago, the mantra was, you can't die from epilepsy, and epilepsy does not cause brain damage. And a lot of this had to do with the movement of science and the movement of advocacy together. And we now know that's not true. And there's a huge genomic and precision medicine movement. And I frankly am cheering you guys on. I hope that you find the best treatments and cures for CACNA1A, because it will mean less LGS. So this has been our journey here, and Savannah is just doing fabulous. She's 28 now. And we know that from the genomic revolution that, you know, there wasn't much progress made from the 60s to the 2000s before the genomic revolution in finding the causes of epilepsy. So these colored graphs up here show we knew these causes up here, and this gray part down at the bottom was all unknown. And now we know that a lot of that is genetic. And we know that there are precision medicines today for specific, you know, go on this medicine for SCN2A, 1 epilepsy, glucose transporter deficiency, GLUT1 deficiency, go on the ketogenic diet, right? We know if you have POLG1 that you don't go on certain medicines, and we also know things to watch for. So it's really, really burgeoning. So in my last minute here, and I know I'm getting close to time, Lisa, but I just want to talk about why it's so important and answer the PhD question. And then I'm happy to take questions if there's time. But to answer your question, do you need to get your PhD to understand your loved one's rare disease? And the answer is no. You're probably going to have to get about 7 or 8 of them. You're not going to go to school to do it. You're going to do it in the life through the school of hard knocks, as they used to call it. And it's because the system is so complicated. And thankfully, there's people out there to help. Like, I'm here to help, and I do have a PhD, but you have all of these doctors that are here and these scientists that are here. So you don't actually physically have to go learn what they know, but you need to partner with them. This is the way that the scientists think about the world. So if you're a basic scientist, you're just thinking about your fish or your flies or your mouse models. If you're a translational researcher, you're thinking about, you know, the clinical trial phase, and preclinical sort of encompasses everything that happens in the animal or in the lab. And if you're a doctor, you think about evidence-based medicine, right? And laboratory studies would incorporate, you know, this early clinical trial and this early research, and then you have all these levels of evidence. But that is not how we as patients think about this journey. This is a journey map, a care map. And so this is one family, and this mom drew this care map, and these are all the school issues, and these are all the life advocacy issues, and the recreational issues, and the legal and financial issues, and the sport issues, and the developmental issues. And here's health. Okay, so this entire timeline, which is the whole world of science, is represented just by the blue train over here, right? If you think of this as a subway map. And we still have the purple train, the red train, the green train, the blue train over here, the turquoise train that we're riding on. And so we need to bring that knowledge in to the physicians. I always wonder if physicians had known about the calcium supplement issue in my daughter, if we wouldn't have thought sooner to look at calcium mutations or to try Varapamil. The other thing is we know the disease the best as patients and families, and we want to bring that information in so we can design treatments that are meaningful, because I'll tell you what, there's a lot of things out there that have been designed for people with LGS that are not helpful at all. And then there have been things that have been helpful, but we also want to push things through. We know from the Institute of Medicine, they wrote a report in 2015, and that it takes 17 years for something to go from science to the patient experience. If you want to speed that up, you are going to have to raise your voice as a community and speed that up. In addition, we know that healthcare is more than just biology and medical care, right? So if we're leaving it to the scientists and the clinicians, they're thinking about biology and they're thinking about medical care. They're not thinking about social circumstances, behavior, and environment necessarily, and nor can they. That's not their job. But we don't get to silo. We as patients and families do not get to silo ourselves in a research lab. That's what I love about I would love to go back to research and just spend my time with my stem cells and my zebrafish doing, you know, precision medicine, because then I didn't have to think about this whole, you know, bigger picture of everything that's related. And you know, we're really, as patients, the only ones that have can really say that it's all about the patient. And I say this with deep respect for all of my academic and industry investment and payer partners, but they have other incentives. Academics have grants and publications and promotions as their incentives, and they are very busy and they are very good at what they do. But we may not we don't measure success and improvement based on grants, publications, and promotions for our partners. We can't do it without them, but that's not how we as families measure whether it's about the patient. And it's same with our industry partners, our investors, payers, people in the funding sector. So we as advocates really need to bring everybody together, convene like you guys are doing, and go from being these passive patients to really being partners, co-designers, and co-producers at every stage. And I just have 2 more slides that I'll show and then I'll stop. But it used to be this paternalistic model in medicine where the doctor said, trust me, I'm a doctor, do what I say. And that was great for that time because all of the knowledge resided in doctors. They went to school and they trained with that. But as the digital age has come forward and as patients have become more educated and as we have access to information, we don't have access to knowledge, but we have access to information on the web and things. We've formed a more informed medical decision-making. And now we want a shared decision-making model. And we don't just want this in the doctor's office, we want this in every aspect of this. We want a shared decision-making model in basic, in translational and clinical, and in evidence-based medicine. And the reason we want to do that is because we will save doctors and researchers and companies time and money. We will do this. I have seen this over and over because we'll advise them on what really matters to patients. And we are the only ones that can advise them on what really matters to patients. So I'll end here with this slide here. I love this slide, and I don't mean to call anybody a dog. I personally would love to be a dog, but it says, meanwhile, at today's Feline Engagement Committee, decisions were made on how to increase feline recruitment, and everyone around the table is a dog. And I just think that the more we can diversify this, the more we can bring in cats, and the more we can bring in mice, and the more we can bring in every group with researchers, scientists, clinicians, payers, the government, regulators, policymakers, the more we are going to solve this problem of these severe rare diseases in our kids faster. So keep doing what you're doing, moms and dads, participate in the natural history study if you can, keep funding research, keep pushing it forward, because I would hate to have a disease where there is not an advocacy organization pushing and convening and uniting and driving things forward. And I just want to do a shameless plug for our meeting of the minds that is coming up, since there's so much overlap. If you guys want to hear some scientific talks about LGS, come join us. Thank you for having me. And I cannot wait to see what CACNA 1A Foundation, when all of you who are behind it, do 1 more year from now, because it's been a pretty great year for you. Thank you.